most most dreaded, devastating, and catastrophic. See the words that has been used. It's the most dreaded, devastating, and catastrophic. Catastrophe means generally this type of terminology we use for cyclones, tsunamis, and all such stuff. So coming to the India, it was first reported in Chinsura in the West Bengal, I think, 1911, and later from there the disease has spread to in Andhra Pradesh, Tamil Nadu, Kerala, Karnataka, Maharashtra, West Bengal, and which are the chief major producers of the banana. So coming to the Panama wilt, this is caused by Fusarium oxysporum, Fusarium oxys, former species cubens. So technically, we'll be writing in a shortcut FOC. So in coming to the Indian variety, in India, this particular pathogen, Fusarium oxysporum, former species cubens, in India, the particular race that is existing is race 1. So this race 1 is highly is these particular varieties which are silk groups of banana varieties. They are highly susceptible. Where previously I told the majority of Indian production is on grand name variety, for which that particular variety is very less infected, or it will bit of tolerant for this particular Panama wilt. So and particular Panama wilt means because of the race 1. So these are the typical symptoms of the race 1 type of banana. Because I'm stressing more about the race 1, because this race 1 is very highly this is very virulent on the Amrutpani varieties or the type of varieties, and it has hardly any effect on the granule variety of the banana. So coming to the symptoms we see, so they basically this Fusarium is a soil-borne disease. Soil-borne means this particular pathogen, which is a fungus pathogen, is residing in the soil. So from the soil, it will enter into the plant system through the roots. So generally, these fungal pathogens, they are very passive. Directly, they cannot enter into the root system. So whenever there is infection in the soil, there are certain nematodes. So when they damage the root system and enter into the plant system, at the time, this pathogen also will enter through the damages, through the microscopic openings or microscopic injuries caused on the root system, and this fungal mycelium will enter into the plant system. Then it invades this is a vascular pathogen, means that it is mainly residing in the vascular tissues such as xylem and phloem. So from there, it enters the root system. From the root system, it enters into the shoot or the pseudostem, which is like a cylinder. It's like there. So there, more the vascular tissues are there. There, it is getting infected. From there, it will spread into the leaves. So leaves, and from there, initial symptoms will be seeing on the foliage as yellowing. So younger parts, they are most vulnerable, and the symptoms you can see yellowing of the leaves. So and over a period of time, when the pathogen is well established in the plant system, we can see this pseudostem outer whorls. We can see the splitting of the pseudostem outer whorls. So as the disease progresses, we can see the cracking. And if we break open the rhizome, which is present in the soil, we can also see this type of purple coloration of the vascular tissues. And if we see this such type of symptoms, if we give a longitudinal cut of the pseudostem, you can see reddish to brown discoloration of the vascular system, because these type of symptoms are very particular for the farmer's field of farmer's viewpoint, because these are the typical symptoms we can for identifying the Panama wilt. Other thing is, over a period of time, we can see at the all the leaves, they will be breaking at the PTO level, and they will be present as a whorl outside the pseudostem. The central pseudostem is there, right? So around the pseudostem, the top leaves, they will be breaking at the breaking at the petiole region, and they will be hanging like a girdle around the pseudostem. These are the typical symptoms. So coming to the causal agent, Fusarium oxysporum, pharma species cubens. So as we already shown the pictures, we can easily understand reddish to brown discoloration of the host vascular system. Vascular system, it is nothing but the vascular tissues, xylem and phloem. So as I told, the first it is seen in the feeder roots, from where the plant get the nutrients. So as I told, it will first through the microscopic injuries made by the nematodes or by during the intercultural operations, there will be some damage to the root system. So through that damage, these spores, these pathogenic spores which are present in the soil or the mycelium, they will enter into the root system. They will enter into the feeder roots, from then into the main rhizome, and particularly at the stele and cortex. where these vascular tissues like xylem and phloem are considered. From it, it will move to the pseudostem and finally to the outer leaf sheath. So external symptoms, as I shown you, we can see the yellowing of the oldest leaves and longitudinal splitting of the lower portion of the outer leaves of the pseudostem. As previously I shown, we can see here like this longitudinal splitting of the central pseudostem out to the outer whorls. As the disease is progressing means the outer leaves one by one layer, it will be getting more deeper and deeper. So as a result, because the pseudostem which is giving the foundation or the support for the plant, that will break and it will completely break down and it will topple at the from the base of the plant. Finally, younger and youngest leaves, the collapse till the entire canopy consists of dead and drying leaves. So initially it will appear as yellowing, so which is called yellowing or the chlorosis. And finally, over the disease progresses, it becomes necrotic, means complete drying of the leaves. So pronounced reddish to brown discoloration of the vascular tissues. So when we cut the pseudostem, because the vascular tissues are present in the root system and also all the living parts. So if we give the cut in the longitudinal cut for the pseudostem, we can see that the reddish brown discoloration. Similarly, for the rhizome also, if we give a cut, we can see this type of discoloration. So over a period of time, because this pathogen is so much dreadful that within 1 to 2 months of the plant infection, if the plants are planted in the field, within 1 to 2 months, it will completely die. So there will not be any flowering or fruiting till that stage also they will not. So there will be decay and topping of the plant. So all this information, as I told, it is these symptoms are like in Indian conditions, the only the race one of Fusarium oxysporum is present. So in other countries like African countries or like Latin America, only tropical race 4 is there. So tropical race 4 is highly pathogenic on the grandin varieties, which tropical race 4 is not present in India for the past more than a century or more than 1 century and around 110 years. But this tropical race 4 has migrated due to movement of the planting material or from due to many other reasons. It has moved from American, Latin America or like South American countries to the Indian, so this Asian countries. So it was reported in identified in Taiwan, and later it jumped from Asia to the Middle East and Africa and reached Lithuania. So this was initially, so outbreak of the FOC or Fusarium TR was first placed in the northern territory of the Australia. So I think like 5-6 years back, it was first reported in the Australia, and from there it spread to the China, the Asian countries, Indonesia, Malaysia, and Taiwan. So in 2016 or 17, the first report of the infection of the tropical race 4 was observed on the Cavendish bonnet, because as I told, this race 4 will not, this race 1, which is present in India of the this Panama wilt, will not infect the Cavendish varieties. But suddenly, to the surprise, all the researchers, they identified that Panama wilt symptoms they are seeing on the grandin variety. So that has surprised. So they have confirmed by various molecular methods that it is not race 1, the tropical race 4 has entered the India, and it is spreading to other parts of the country. So the ICR, Indian Council of Agriculture Research, and other research institute Tamil Nadu, they have taken on a war footing, and they have particularly they were successful in limiting this particular race, which is tropical race, only to the Bihar and Uttar Pradesh. So as it is reported, it is first hotspots. Hotspots means the particular areas where the disease is very severe in proportion, or where it is rapidly spreading. Coming to this, was reported as tropical race 4. It is considered as one of the most destructive of all the plant diseases by the Food and Agriculture Organization of the United Nations. So this has the capability to geoparadize or the $26 billion banana trade. So this is the information about the tropical race 4, in which in various newspaper they considered this particular tropical race 4 as COVID and for the banana plantation, and it can threaten the $1 billion industry of the Indians. So now banana COVID hits plantation. So this is in Hindustan Times, and India in a race against wilt in the Cavendish banana. So similarly, just before it had entered the India, it was reported in the Australia. So where the banana industry is also very much important, and there the banana industry is valuing almost $600 million. So as we are all familiar, the quarantine means we started hearing only from the COVID, but this particular quarantine is a terminology particularly used in the plant pathology, where we isolate certain diseased plants we are observing a new diseases, we restrict the movement of the plant, the such type of diseased material from the infected areas to the new areas. So whenever when we immediately after new Queensland territory of the Australia, they identified this disease and they confirmed molecularly. They put it as a quarantine and they have restricted. They did not allow the any personnel to enter into that infected area, and also either from the rhizomes or even the soil particular area to the other adjoining banana growing areas, because this is a soil borne accidentally if it is spreading from one area to the other state, other districts or where this pathogen is not present, means it will have a huge effects. So a discovery of TR4 in Australia's banana growing, it has threatened the $600 million industry. So it is a plant pathogen. So we need not worry. Though we say COVID of the bananas, these particular pathogens, they don't have any effect on the human beings. They causes no health risk to the humans. The disease attacks only the plant varieties, banana varieties, including the commercial Cavendish, which accounts to 95%. So India, it is only Cavendish, it is only 70%. But in Australia, it is 95% of the production of the varieties are occupied with the Cavendish. And this tropical racefour of the particular has been moved from Latin America and other places to the Australia. So recognizing the importance, the Australian government has invested $10 million exclusively for devising the management and for preventing the spread of this disease from the Queensland to the other areas. So coming to the disease management, so generally effective chemical control measures do not exist. So even after more than a century, the effective control measures, they are not existing. The only thing is we should collect the using a healthy plant material which does not have any disease, because most of the plant we time the farmers use the suckers. So these suckers, what we suggest is we should use the suckers which are not from a healthy plant, which have given from a healthy plant and not from the infected plant, because infected plants have the property pathogen, and if we are collecting the suckers from the plant from the infected one, we are spreading the disease from the infected area to the new areas where the disease is not existing. So limited success is there with the rhizome injection, means where we dig out the rhizomes of the banana and we make a small hole and we pour 0.2% carbon dioxide, means 2 ml per liter around. We'll make a small hole or like in the banana rhizome and we'll pour around 6 ml like that. So that will penetrate into the rhizome, and if any pathogen is there, this Fusarium pathogen is there, that will kill the pathogen or it will reduce the effect. And another thing is, best thing is, if you are observing this disease initially, the best thing is you completely remove the plant and discard it along with the soil up to 2 to 3 feet, either horizontally and also vertically, because that also contains the pathogen. Avoidance of the ratoon cropping, means in farmers what they see is generally they plant a banana, and after certain point of time, after one season, they will harvest and they will allow the suckers of the plant to again. So if you are observing a disease, we should not allow the ratoon crop, means suckers which are from the plant from the same area, we should not allow the suckers to grow. So means we have to completely remove and we have to start with we should either, if the disease is severe, we should go for another alternate crop which is not infected with any other alternate crop, or we should give a crop holiday for this particular, so that will reduce the amount of inoculum present in the soil because of the absence of the host. So TR4 management strategy, as I said, it's a severe dreadful disease. So the best thing we can do is we have to only identify the cultivate the disease resistant varieties and soil health management practices. Soil health means we need to say there are soil health means there are certain beneficial microbes like trichoderma which are identified antagonistic to this particular. So if we multiply those particular beneficial microbes and we make the formulation, we add to the soil, that will arrest the growth of the pathogen. And cropping system and agronomic practice. Agronomic practices means suppose infected material is there, we have to completely destroy. So we have seen that the diseased portion, such as like infected leaves like that or the pseudostem, immediately we have to, this after crop harvest, we have to collect all the plant the dried material all that, and we have to burn it off. So because if we don't burn it off, the pathogen will still enter into the soil. So this is called phytosanitation. Healthy planting material, biological inputs, soil health oriented strategies, agrobiodiversity. Biodiversity means we have the habit of cultivating the same variety in a several thousands of acres. So if the particular variety is susceptible to important dreadful disease, all the varieties will get infected. So we have to grow multiple varieties with varying levels of resistance. So even some varieties are getting infected with this and getting infected, and there is a loss. Even then, because of the biodiversity, we are able to get from the even from the other varieties which are less infection. So a little bit of success is also there from the ICR, where they have developed a certain formulations called Trichoderma ricci. So it is a soil. This particular biological control agent is present in the soil, Trichoderma ricci. From there, it was isolated and it was tested against the pathogen in the laboratory studies, where it has given 85% control of the 85% inhibition of the pathogen fungal mycelium. So it not only under in vitro studies or laboratory studies, when this particular biocontrol is also applied to the healthy plants or the rhizomes, when and simultaneously if we are infecting the plants with a particular pathogen, this tropical race 4, then the even after even though the pathogen is present, still the plant is able to grow properly and also give yield. So that indicates, upon challenge inoculation with the pathogen and simultaneous inoculation of the biocontrol agents like Trichoderma ricci, which is highly antagonistic, the pathogen is not able to perform properly or is not able to infect properly, and still we can able to harvest the and there is a healthy parameters like even though it is infected, the plant growth is proper. It was able to shoot, it give the shooting, means a flower formation and the bunches formation. So as per the ACRP fruits recommendation, All India coordinated of the ACR, so these are the certain management practices: use of disease free sucker material from the disease free yield, and dipping of the suckers before planting in the carbendazim 0.25% for 45 minutes, followed by drenching with carbendazim in 5th, 7th, and 9th month at 0.2%. Means we have to prepare the solution 2 ml per liter. And again, carbendazim injection, as I said, 3 ml of the 2% solution at 3, 5, and 7 months after planting, and application of lime per pit. Here I am saying it is lime because this particular fusarium, it grows rapidly or it flourishes well under acidic pH, means 5.5 to 6 or like 5.25 or 5.5 to 5.7. So when the soil pH is 5.5, this pathogen is growing luxuriantly in the soil. So if we alter the pH, so into the alkaline medium, so like how we can increase the pH means, if we add lime, little bit of lime to the soil, it increases the pH. Here this lime is not directly killing the pathogen, but it is increasing the pH of the soil. At that increased pH, the pathogen is not able to flourish. So next is this is one of the important, so I have given elaborate explanation about the Panama wilt. Next is another important dreadful disease, leaf spot diseases or foliar diseases. So it is a general common terminology. Whatever leaf spot we see, it is the whether scientists or even the farmers, they call Segatoka disease. But but there are many foliar diseases which are causing leaf spots on the foliage, like Segatoka leaf spot. And it is at this point of time, I want to say Segatoka is also two types: yellow Segatoka and black Segatoka. Yellow Segatoka is present in the Indian condition. In the African condition, there is a black Segatoka. Though the species is, though the pathogen, though the genus is separate, Mycospirilla is there. So in Indian condition, yellow Segatoka is caused by one species, and black Segatoka is called by another species. Similarly, there are other symptoms such as other pathogens such as Podana, Diatoniella, and Cladosporium speckle. Coming to the yellow Sigatoka, it is caused by the Mycospirilla musicola, because it is called as a Sigatoka disease because it was first reported in the Sigatoka Valley in the Fiji Island. So it is as we know, these are foliar diseases. These diseases, they cause leaf spots, and and they reduce the photosynthetic area. When there are more leaf spots are on the foliage, that reduces the photosynthetic efficiency of the plant, because when there is a proper photosynthesis, there are more sugars produced in the foliage, and those sugars are translocated to the fruits. And there we can see, if there is no disease of the foliage, then we can see maximum very good size of the bunches, and also optimum more number of bunches, more number of fruits, and very good size of the fingers or each fruit. So when there is a photosynthetic area is reducing, that will affect the number of hands produced, the size of the fruits, and the size of the bunches. So as much as if we are able to reduce this foliage, we can able to, that will increase the quantity and the quality of the fruits. So the initially what we see is when there are foliar diseases, we can see initially small spots are like that. Where the disease progresses, these spots progresses, and they will completely, we can see the necrotic portions on the foliage, and that is related to the premature death of the large areas of the leaf tissue, reducing the photosynthetic capability. So these are the different symptoms of progression of the disease. So on the leaves, there are secondary veins. So initially what we'll see is there will be pale yellow streaks. In between, there will be slight discoloration in the initial sites of infection, slight discoloration. As the disease is progression, in between the secondary veins, we can see either pale yellow streaks or brown streaks. As the still if more disease is progressing, we can see elliptical spots with central grayish portions, and these lesions, they will be surrounded by yellowish region or a chlorotic regions. So when there are this type of spots or many are there, so one spot will merge with another one, which we say coilis, and we can see large areas of the necrotic portion or drying of the leaves. When the leaves are drying, as I told, it will reduces the photosynthetic efficiency, and that is reducing the production of the more this necrotic portion. Because of the photosynthesis, photosynthetic efficiency is reduced, and that is reduction in the sugars, and that is reflected in the reduction in the size of the bunches, fruits, and like that. So as I told, and the other is the chordana leaf spot. So that is caused by the chordana musae. So this first appears as a tiny yellow spot, which is later characterized by oval or eye-shaped spot with grayish center having faint concentric zonation. Another leaf spot is the diatoneal leaf spot. It is caused by the diatoneal turulosa. So how we can identify is on the foliage, we can see from the margins of the leaves, we can see inverted V-shaped lesions. These are the typical symptoms of the sigatoka. Say where we see this type of lesions, where we can see the elliptical spots surrounded by yellowish halos. So how we can differentiate in the field level means these are the types. So first one A, all the disease symptoms are yellow sigatoka. Second one is a B, so where we are seeing that elliptical spots with grayish portions, that is caused by the chordana leaf spot. So the third one is the inverted V shape from the margins of the leaves, that is called the diatoneella. So these many symptoms, these are some of the, as I said, these are the three foliar diseases. Along with this, there are other leaf spots also like Philosticta and also cladosporium. But the farmers, what they say, only one leaf spot, sigatoka disease. So there are different types of pathogens causing same type of disease, but with the varying all the foliage. So each pathogen is different, and each pathogen for controlling the chemical is different. So we have to use different types of strategies for management. So this is the cladosporium specule. So most all these foliar diseases, most of all these diseases we can see particularly under the rainy season, when these plants are planted very nearly when they are densely planting, because there will be more transpiration and there is more piling up of the humidity in the phyllosphere of the plants. So when there is more humidity, low temperatures, at that time these foliar diseases occur very prevalently. So how to manage means clean cultivation. Clean cultivation means, as I said, these type of symptoms are there. So immediately after the harvest of the bunches, all we have to collect when there are symptoms, they will be having the pathogen or inoculum. So we have to destroy immediately after the harvesting of the plants. We have to collect all these debris, plant debris, and we have to burn it off. If we burn it off, the pathogen inoculum gets reduced. Otherwise, this inoculum gets mixed with the soil, and it will spread to the, again we are planting the banana in the next season, it will cause infection. So removal and destruction of the affected leaves, keep the banana field as weed free, and remove the sucker timely. So avoid planting at close spacing, because more the plants are spaced very nearer to each other, that increases the humidity and increases the disease. So provide proper drainage and avoid waterlogging. So, and the other thing is, we have to, as we told, it is very difficult at the field level to independently separate which disease, whether it is the sigatoka or chordana or diatoneella. So we have to use multiple number of fungicides as alternatively, because there is no surety that only one type of disease, multiple diseases, multiple leaf spots are there. So if you are using different molecules like carbendazole, propiconazole, tridamorph, alternatively. So we can expect a very efficient control. These should be mixed with, because once we spray on the foliage of the banana, it will not stick. It will, because it is a waxy type. It will come fall down. So we have to add some sticking agent. As per the ACRP fruits recommendation, it is called 3 sprays of propiconazole, means 0.5 ml. Generally, propiconazole we use 0.1 ml per liter, or 1 ml per liter, or 0.1%. So there is a new recommendation is that if we are able to add some petroleum based mineral oil, so petroleum based mineral oil at the rate of 10 ml per liter, then we can reduce the dosage of the fungicide by 0.5%, by 50%. So instead of 1 ml per liter propiconazole, we can use only at 0.5 ml per liter. Means for 10 liter tank, just 5 ml is sufficient. Along with 10, we have to add petroleum based mineral oil. Generally, oils don't mix with water, but these type of mineral oils, they are water miscible. So these fungicides easily get mixed with this water, and also petrol oil, so it will mix. And besides this, when we spray this 10 ml per liter of the mineral oil, these mineral oils, they will form a layer on the surface of the foliage. So when there is a mineral oil, then if there are any pathogenic spores, they will not be able to germinate because of the petroleum oil. So when there is water, the spores will germinate and enter into the plant system. But because of the presence of a layer of this mineral oil, the pathogen will not be able, spores will not be able to germinate and enter into the plant system. So other recommendation is 3 sprays of propiconazole at 30 days interval, along with some biocontrol agents are also there. Those formulations like Bacillus subtilis or Pseudomonas fluorescens are also effective. Am I audible, madam? Hello? Hello?
Yes,
Hello,
sir.
am I audible?
We are audible.
Okay. So this is a very good recommendation, because we are able to reduce the dosage of the fungicide by 50%. So this recommendation, because I worked in the research station and also the colleges, and so we popularized this technology through our final year project students in a very good in Guntur districts by conducting frontline demonstration, where we used this because farmers are unaware of this type of practice of using mineral oil. So we popularized this all India coordinated research project along with our college administration, and it was very good, rapidly adopted. It was very much adopted by the farming community, which very big progressive farmers we had, and this was covered in the several newspapers, our activities. So these are the fungal diseases. Coming to the bacterial disease, we have very important disease like rhizome rot or tip over. So this is caused by bacterial disease, Erwinia keratovara. This is also importantly dreadful, like the fungal disease like Panama wilt. So this is seen in the Andhra Pradesh, Tamil Nadu, Kerala, Karnataka. So as I told, this rhizome rot is severe on the dwarf Cavendish and Telachakravelli variety. So this is also like Panama wilt. This is also soil-borne disease. The bacterium resides in the soil. So at this point of time, we need to say that rhizome rot, this particular bacterium is a little bit of thermophilic. Means this bacterium loves a little bit of higher conditions of temperature. So the pathogen will be present in the soil in the all through the year, but from June to March it will be dormant. So whenever there is summer season, when there are higher temperatures, the soil, the temperatures increases. At this temperature, elevated temperature, the bacterium becomes very energetic and it starts causing infection. So the very important, we can say that rhizome rot or tip over disease is very much prevalent, or it assumes severe proportions under summer conditions. So these are the typical symptoms. We can get confused. These symptoms are they are similar to the Panama wilt. So we can see here also yellowing, and we can also see this is also pseudostem splitting, and the central whorl of leaves that will completely dry off. This, why I am telling this is central whorl is dying off, means because this central whorl young leaves, this will be harboring the inflorescence, because the economical part in this banana is inflorescence. From there the flowers and fruits will come. Whenever there is infection of the rhizome root, the central whorl will die. Means there is no flower forming, there is no flower setting, and there is no fruit setting, and the plant will die. So this is the differentiating symptom of the Panama wilt in this one. So at the soil region, if we dig, we can see the rhizome, we can see this type of rotting. This is the typical symptom of the bacterial rhizome rot. That's why it is called as a rhizome rot. When we see the Panama wilt, we only see that pinkish reddish brown vascular discoloration. But here we don't see such type of discoloration. This will be rotten mass of the rhizome. The rhizome which is present in the soil, that will get rotted, because this particular bacterium, it releases certain enzymes like cellulases and pectinases that will degrade the cell wall. So what are the symptoms? This already I shown this type of symptoms, but little bit of explanation. Rotting of a newly planted rhizome that failed to sprout, stunting and yellowing of the plant. So dark brown necrosis appears in the lamina. Pseudostem tips over. So it tip over means that it will break at the collar region, at the soil region, and completely the plant will topple. As a result of this, the plant will produce small sized fruits or less number of fruits with poor quality. And in case of this, if the disease is present in the from the initial conditions, within 3 months, the entire plant will die. There will not be any flower formation. There will not be fruit setting, and the entire duration of the plant will not be seen. So toppling of the matured fruited plants. So how to manage means selection of the rhizome. So here also, just like other diseases, it is a soil-borne diseases. So we have to collect the healthy rhizomes from the uninfected fields. So from the healthy plants means healthy rhizomes, so there is no disease. So planting in the well-drained soil. So and also avoid planting during heavy rain periods. And because it is a bacterial disease, we give for a COM injection with a 0.1% streptocycline, and along with that 0.2% carbendazim. Because the soil has both Panama wilt and bacterial disease, we give both. We use both the chemicals, streptocycline for as a antibacterial, antibiotic, and carbendazim for controlling the fungal diseases. So another thing is bleaching powder. So this is a very common, everyone knows this bleaching powder. So if we are able to add that a rate of 25 grams per plant solution, means for 1 liter, we add 25 grams and prepare the solution. All we have to do drenching at the base of the plant. So this bleaching powder, as it says, bleaching, it will completely bleach off the cells. It means whether it is pathogenic microbes or like useful microbes, all they will get killed because of the planting. So another is farmers practice. All these people like farmers, they are going for a cow products. They prepare panchagavya. Panchagavya means cow milk, cow curd, cow ghee, and cow milk, cow curd, and cow urine, cow dung. All this they will mix, and they will allow it to ferment by adding some molasses like that. So under that fermented conditions, some beneficial microbes will grow. So that fermented conditions, that such type of panchagavya, if you add to the base of the plant, that will reduce the disease incidence. So, and this ACRP recommendation is that we have to add streptocycline 1 to 2 liters per plant at the rate of 500 ppm. And as I told, the soil, when the soil is getting heated up because the elevated temperature, the disease is increasing. So how we can reduce the soil temperature means that is not in our control. But in between in the banana plantation, if we are able to grow the intercrop with the cover with a cowpea or a sun hemp, so it will be acting as it will cause a smothering effect. It will completely cover the soil in between the banana plants. So that will reduce the sunlight, or it will give a shielding effect. Because of that, the soil temperature doesn't increase very much even into the summer conditions. So this type of agronomic practices, if we are able to do, that will reduce the incidence of the disease. So this is and 6 grams of bleaching powder up to 4 months of planting, and these are the recommendations. So this is the important bacterial disease, as I told, Moco wilter, caused by Ralstonia solanacearum. So initially, this is not present in India, but every agricultural university student is very much familiar with this disease because it is part of the syllabus. And everyone feels that this disease is there. But practically, this disease is not present in the current Indian condition, although there are some conflicting reports that the presence of the disease. But officially, it is not yet confirmed throughout the country. So even, but in spite of that, we can cover this one, Moco disease. Initially, it was considered as Bacillus musae. So later it was modified, it was renamed as Bacillus solanacearum, and it is later confirmed as Ralstonia solanacearum. So it is a bacterial disease. So how it is spreading means, so if there is infected plant, if you are using some intercultural operations like a drying of if we are removing the dried leaves, at the time if you are cutting the dried leaves means the plant system has bacteria. Those bacteria from the cut ends, they will stick to the sickles or any other agricultural implements. If you are using the same agricultural implement for another intercultural operation for the healthy plant, the bacterium will enter into the healthy plants. So through the wounds or some natural openings, they will enter into the plant system. So these bacteria, they will be able to persist in the soil up to 1 to 1.5 years. So these are the different types. So because this disease is not present in much in India, so I'm not covering much. Just the 2-3 minutes, madam. So, but these are the typical symptoms of the Moco wilt, Ralstonia solanacearum. So these are the typical symptoms we can see on the pseudostem, foliage, and we can see this type of rotting or blackening. So this is the thing. So with this, I end the presentation. If you have any doubts, you are most welcome.
Thank you, Dr. Sesha Kiran, for a very detailed presentation. I'll take up the questions. In the meantime, you can stop sharing the screen and you can on your camera, please.
Okay.
Yeah. Okay. Yeah. So the first question is.
Is my voice audible, madam, or you
yes,
have any
you're
problem?
audible. You're audible. It's clear.
Okay. Thank you, sir.
How frequent are fungal and bacterial diseases in banana?
Yeah, it is a frequent disease. Like we can say that as it exists with the crop. So whenever there is a crop, there is a this particular fungal and bacterial diseases. As I said, Panama wilt, it is a very dreadful disease. It is almost present in majority of the plantations. Even fungal disease, I said, it is spread through airborne. These are like Mycospirilla, Muse, or Cigatocalli, Spot Cordana. They are present in the foliage. So whenever there are rains, there some water will fall on the infected leaves on the spots. So the drops will fall. The drops will fall on the infected areas, and it will bump onto the healthy portion. So along with the drops, this pathogen also spreads to the neighboring areas. So it is almost whenever there is a crop, there is a pathogen. They coexist together.
Okay. So next question is, what precautions farmers can take to avoid these banana crop diseases?
So as I told, the type of management practice depends on the identification of the correct pathogen. If see, because this Panama wilt and rhizome rot are there, so the symptoms will appear very similar. So how we should be able to differentiate whether it is a Panama wilt or a bacterial rhizome rot. So if you go symptom wise, so if there is a rhizome rotting, means it is a bacterial rhizome rot, because the yellowing is common in the rhizome rot and also Panama wilt. So I have given a elaborate management practices given by the ICR and also the university level. If we are able to critically analyze based on the symptomatology, accordingly we can go for a recommended practices. So these are limited. These are very much dreadful diseases, but still with good agronomic practices and also recommended practices by the ICR and traditional university, agricultural universities, we are able to reduce the incidence of the disease. Unfortunately, these diseases are very much dreadful, and they are present since ages, and 100% success is not possible.
Okay.
We can only reduce the disease incidence, and we can only enhance the crop duration and increase the quality and quantity of the crop. Though not, we cannot completely avoid the disease.
Okay, I'll move to next question now.
Yes.
What is the current focus of research on fungal and bacterial diseases in banana?
Yeah, all the Indian institutes and state agricultural universities are very much focused on this particular disease since long time. They are continuing the research, and they are coming with new methodologies and recommendations. Depending on the earlier, it was only say bleaching powder. Now means new streptocycline and a combination of management practices. Earlier we said only streptocycline they used to say, or a bleaching powder. So we are trying to know more about the disease. Means it is becoming severe. That is epidemiology we call it. So it is under increase, the disease increasing under summer conditions. So how we have to manage, not only through chemicals, agronomic practices. We say that in between the banana crops, in between the banana plant, if you are able to cover the soil with other intercrops like cowpea or sunhemp, that will reduce the disease incidence. So this type of new experiments are going on, and we are giving a new recommended practices for effectively reducing the disease incidence. So earlier this palm was Panama wilt. So they used to give only clean cultivation, phytosanitation, and spreading from reducing the movement of the inoculum from the infected areas to the other areas. So now we are giving a rhizome injection, we are and other improved practices, so that we can new recommendations are coming up, replacing the old recommendation, so that we have to can effectively reduce the disease. So we said, what is the status of the research? So earlier we were not aware that this tropical phase, this tropical phase 4 is not present in India till 2016. We have been coexisting with the banana from some several centuries. We feel that only race 1 is present. So we identified in 5 years ago, we have identified this tropical phase 4, which is not present in India or Asian countries. This is entered into India. Accordingly, the universities, this scientific staff of the universities, and also the ICR Institute, they have responded on a war footing. They conducted extensive research, and they identified it molecularly, morphologically, and developed the recommended practices, not only from the National Research Center for Banana, but even for other institutes, even in the North India, which where their mandate crop is not banana. They also worked on this disease to develop a very good effective management, because this our country is the largest producer of the banana, and if this disease is spreading from hotspots from Uttar Pradesh to other states like Andhra Pradesh, and it will have a severe impact. It will affect the banana industry. It will affect the banana farming community. So that's why it was called as the banana COVID. So because of this responding to this call of the tropical race war, extensive, because of the extensive research, they were able to curtail these diseases. Because of
Okay.
their practice, though it is a 5 years, it did not spread from. There was strict quarantine imposed of preventing that infected planting material from tropical race war from the infected areas from Bihar and Uttar Pradesh to the other areas. Otherwise, if we are not if these quarantine measures were not followed, means it would have entered into the Andhra Pradesh, it would have entered into the Kerala, and it would have spread like a wildfire. So research is
Okay.
there, effective research was there, and extension personnel were also there. Because of the marketing management practices, they were able to arrest this disease or limit the disease only to the infected areas.
Okay. I'll move to next question. This is a slightly different question.
Yes.
What drives you as an agriculture research scientist?
What is it? What?
What drives you as an agriculture research scientist?
Like what are the drivers? Means, everyone in I am also a student like everyone who is pursuing. When you are studying in the undergraduation, certain disciplines will be more interesting to you. That is the time when you are in a master's or a batch. When you are in a bachelor's, you feel very much inclined to certain areas, or some areas fascinating. So that type of interest, if you work on that, so I want to work on a plant pathology, so or biotechnology. So at the time, we should see, we should explore the opportunities, what type of research or which universities are going conducting that outstanding research. Accordingly, we have to devise certain plans, select for and prepare well for the national level junior research fellowship exams, do good research under a proper guide, good guide. We come out with publications and continue higher educations. So the passion, basically, it is a passion and interest, not for a job.
Okay.
That is,
Next question is like we have Ebola and COVID in human viruses. In terms of plant viruses, which are the most dangerous viruses?
viruses, I have given extensive talk on that. It depends on the type of the crop. So tomato, number of almost 125 viruses are there. Chilies 125, more than 130 viruses are there. Like that depends on the crop. Where a banana is there. Banana, we have banana vegetables. Black mosaic virus is there. Cucumber mosaic virus is there. Banana streak virus is there. So it depends. Like coming to the chilies, chili leaf curl virus is there. Tomato leaf curl, cotton leaf curl. Particularly, we are residing in the tropical conditions. So here there is more incidence of the white flies. So the white flies act as a transmitters or a vectors for bigomoviruses or a Gemini viruses. So here in Indian conditions for the agriculture crops and horticulture crops, this white fly transmitted Gemini viruses or this group of bigomoviruses, they are very much important and they are more dreadful. even whether it is a tomato or whether it is tomato or a chilies or a cotton or multiple crops. But these causing symptoms like these particular bigomoviruses, Gemini viruses, they are much dreadful.
Okay, I'll move to next question now.
Yes.
Who are some of the leading plant virus researchers in India and around the world you admire and why?
Oh, who are the leading virologists? Means we have I have worked under my pursued my PhD with in the Indian Agriculture Research Institute under Dr. Bikash Mandal. I think currently he is the in charge of Advanced Center for Plant Virology. He's a very good scientist. As I told, he's a whitefly transmit. He works exclusively. He works very much on the whitefly transmitted viruses, these Gemini viruses. So I trying to be a good researcher under his guidance, and under very good scientist. We can say like father of Indian plant virology, like Professor Anupam Verma is there, Dr. Paul Kurana, who was a Central Potato Research Institute, and spices in Indian Store Spices Research, Dr. Ishwar Bhatt is there. He's doing some very good research on viral disease infecting the spice crops. Similarly, in the citrus, Nagpur, we have Central Citrus Research Institute. We have its director is Dr. D.K. Ghosh. He's an outstanding virologist. So like this, as I told, it's very difficult to work on viruses. So, but if you are
Okay.
able to work passionately, this is the field that gives you more recognition and also contribution if you work with the passion. So
Okay.
majority of the universities, they have fungal pathologist, bacteriologist, but virologists are very few and they are like considered as diamonds, very valuable.
Okay. Okay. What
Hope I
are
have
the next
answered your
question?
question.
Yes,
Yes.
you are answered. Okay. What are the next question is what are the challenges involved in taking all these good research to the actual farmers?
Yeah, it also depends. Yeah, I told it is not only developing a good technology. These technologies have to reach the farming community. That is called lab to land. So that depends on the effective extensional personnel, like who are able to effectively communicate the about the management practices. See, we have very good management practices developed at ICR level, at Indian Council of Agriculture Research, along with the several state agricultural universities. Out of a dedicated research for several years, like decades, they come out with a very good recommendations for multi-location testing. So unless it is not only important to just develop these management practices, we should have a very good extension personnel. So every researcher, it is not every scientist or faculty who is working, he should do research, extension, and teaching. So unless if you want to teach, we have to prepare well advance. So you have to update yourself. If you are confident, then only you can speak before the students. So he should act as a teacher to update the knowledge, and parallelly he should conduct the research and develop a good recommendation, and then finally he has to do a extensional personnel. So whatever the technology he has it was developed, that should be able to reach the farming community and reach the farmers and communicate to them in their local language, in their understandable language. So every research, every university, the faculty, I feel is that you should be involved in research, teaching, and extension.
Okay. Yeah, I'll just read out two appreciation messages from our participants. One is from
Okay.
Mr. Venkateshappa. It reads
Hi.
as follows. Thank you, sir. Very extensive presentation. You have clearly clarified all our doubts. And Mr. Venkateshappa is asking, when can I call you? Can we call you whenever we want?
Yes, yes, no problem. I have
Yeah.
already shared my number. If you again, I'll share. I can
Yeah,
use.
shared your contact information
Yeah,
with our participants.
97175 97175 80195. So this is also linked to the WhatsApp. So you can communicate to me whenever you need any technical help. And along with that, I have another number 94409 94409 33366.
Oh,
94409 333 So I am available all the time on these two numbers. So if you need any technical help or guidance, if you are a student community, so you need any guidance, as you asked, who are inspiring role models like that. I have a hobby of helping majority of the students across the country, and I have guided few students, and I have helped many students to go abroad. Even my own MSc students, I have guided. She has succeeded in. I have helped her in bagging several international scholarships, and she has started her PhD in Canada with a record scholarship of $30,000 with $50,000 scholarship. So I have a passion of teaching, research, and also assisting the student community so that they can become future researchers. And if I can be of any service to help to them, I'll be very much glad to share my knowledge and mentor for reaching their goals.
Okay,
That gives good satisfaction for me also.
okay, it's very good to know and very nice of you, sir. So now I don't see any further questions from our participants. So now we have come to
Okay.
end of question round.
Yes,
Agricultureinformation.com likes to thank Dr. Sesha Kiran Kollipara for a very detailed presentation and answering all the questions in depth. And we also like to thank all the participants for joining this meeting. The meeting will now be closed. Have a good and safe weekend.
thank you so much, madam, and all the organizers for giving me this opportunity. Again, I gave a talk in January 20th. So I was able to connect with all the people, fraternity or for farming community or the students or entrepreneurs through this platform. All the people, if they need any help, technical guidance or the student community, you are most welcome. I'll be able to, I hope I will be able to meet your requirements.
Sure,
Thank
sure.
you.
Your contact
Thank you so much.
information is being shared.
Yes, sir.
So participants will reach out to you.
Okay. Thank you so much.
Thank you. Thank you.
Welcome.